what are the functional consequences? We've done cellular electrophysiology, and back then we weren't making animal models. We never made an animal model of hyperkalemic periodic paralysis, but someone else did, and they're doing in vivo studies. So there's, any discovery you make or we make, when it gets published, it also engages the interest of others, and there's still lots to learn, I have no doubt, but we've moved on to other projects and haven't been working on that since. But they clearly have functional consequences, and the nature of the consequences are different depending on the phenotype that is, results from different sodium channel mutations.
Thank you. I just have one last question.
Yes.
Throughout your lecture, you mentioned about migraine. So out of my curiosity, I also have migraine. So there are two things that triggers my migraine. One is if I oversleep, if I sleep more than 8 or 9 hours, if I get after 8 a.m. in the morning or 10, or if I miss coffee or tea in the morning. So these two things triggers my migraine. So as you showed about some ion channels, so can you give me some idea, is this because of the alteration in ion channels that triggers my migraine?
Yeah, good question. I don't know the answer. I would predict that there are probably hundreds of genes in the 7 billion people on our planet. I would predict that there are at least hundreds of genes that contribute to your risk or my risk or her risk of having migraine. In the rare families that have a single, with the Mendelian disorders where it passes from generation to generation, that is the result of a single mutation in a single gene that is sufficient to cause the disease. But imagine, for example, 10 migraine genes that we don't know about yet, and each one of them has a polymorphism that has some subtle functional consequence. And then you shake the dice, right? How many of the ones that are kind of leaning toward migraines does my dad have? How many of those does my mom have? How many do I inherit? Because, it's a flip of the coin with every, haplotype, right? And so maybe your mom and dad don't have migraine, but he had 5 and she had 5, but you got 8. You got, 3 from dad and 5 from mom, and 8 is enough to cause migraine in you. So not all migraine is going to be a single gene disorder. And we don't, the familial hemiplegic migraine results from a calcium channel mutation, but that explains a very small proportion of migraine, very, very small fraction of all migraine. I'm not aware of I'm not a big fan of GWAS studies, but I don't know, I don't think that the GWAS studies have pointed to that calcium channel locus as contributing to migraine in the general population. But again, the more genes that we and others clone that cause migraine, the more, every story is like a jigsaw puzzle. And so with the big families that are easier to work with, easier to clone, those are sort of like the corner pieces, right? You can identify those pretty quickly, and then you can look at the edges, and then we have to fill all the pieces in the middle. And as we get enough pieces in the jigsaw puzzle, and we understand what the functional consequences are, then a picture begins to emerge, and that's going to take a long time for complex diseases like migraine. But the rare Mendelian families are going to be critical to identifying those corner pieces and edges, I think.
Thank you very much.
Yes, sir.
Hi. My name is Öner from Department of Life Science. I would like to ask migraine as well. So what kind of effect of tea, coffee, alcohol on the migraine?
Yeah.
How can we trigger migraine or attack like that?
We don't know. I don't know. But in the case of paroxysmal non-kinesiogenic dyskinesia, which is super, super, super rare, those patients have a very high incidence of migraine. And so Shen Yang, who had training in Taipei in pharmacology, said, well, coffee and alcohol are both non-specific adenosine receptor antagonists. They're dirty drugs. They do a lot of different things, but they are clearly adenosine receptor antagonists. And so he used, in the mouse model for PNKD, he wasn't looking at any migraine-like phenotype, but just looking at the movement disorder, he showed that he could induce the movements with adenosine 2A receptor specific antagonists and dopamine D2 receptor agonists. And you may know that the medium spiny neurons in the indirect pathway of the striatum are positive for A2A receptors and D2 receptors. And these mice have abnormal dopamine signaling. And so we know that the abnormal movements are being transduced through the indirect pathway of the striatum based on Chen Young's work. But there are also dopamine D2 and adenosine 2A receptors and adenosine 1A and dopamine D1 receptors in other parts of the brain. And I wouldn't be surprised if some of those other parts of the brain were contributing to migraine via some effect of adenosine receptor antagonism. But as I mentioned, caffeine and ethanol, are very dirty drugs, and they also touch other and modulate other receptors. So I'm sure it's not going to be all about adenosine, and although I wouldn't be surprised if that was a contributing factor to some migraine pathogenesis.
Thank you, Professor.
Right down here. Oh, okay. Yes, ma'am.
Dr. Louis Ptáček, I learned a lot. I learned a lot from this high-end lecture. And my question is, in the pattern of the action potential on the membrane, is there any specific pattern of excitatory change among these patients of episodic paroxysmal disease?
Is there any change in the
On
excitability?
the action potential of the membrane of the neuron?
Oh, absolutely. And again,
Specifically on this episodic disease.
the ones that have ion channel mutations specifically. So we know, for example, in Anderson-Tawil syndrome, which is caused by mutations in the inward rectifying potassium channel KR2.1, that we know from other people's work that KR2.1 is important in the cardiac action potential. The upstroke is sodium, then there's a calcium plateau, then there's fast potassium currents that bring the membrane potential down, and the very terminal phase of repolarization is caused by inward rectifiers like KR2.1, 2, 3, and 4. And we know that that is affected in patients with that particular mutation. All the work that we've done in the sodium channel variants has been in a heterologous system where we can't, which doesn't recapitulate the morphology of the skeletal muscle action potential. But in other people's work, there clearly are changes in the action potential. I don't think that kind of work has been done for the brain channels yet. But there's no question that some of it is going to affect the actual kinetics of activation, inactivation. And then there will be other disorders like PNKD and PKD, which clearly have electrical signaling abnormalities in the brain, but it's probably not so simple as a single, thinking about a membrane in a single cell. When you get into the brain, it's, orders of magnitude more complicated than muscle. You have to think about the single neuron and what its action potential might look like and its firing pattern might look like. But you also have to think about the circuits and all the connections. So, I don't even know how to begin to think about that. It's when you move from muscle into the brain, things get a lot more complicated, and it's going to take a long time to understand that in more detail. Well, because there's a break, let me take a chance to say happy birthday to our young friend in the 6th row, who's turning 12 tomorrow.
Oh,
so
yes.
Have a great day tomorrow.
生日快乐！我for the 这边有一位。
Thank you for a nice presentation. I have a question regarding the PNKD. Since the synaptic protein or the exocytosis is involved, is any chance the dopamine or any other neurotransmitter might be involved in this disease?
Absolutely. So I don't know the full answer to that question, but Shenyang and then with the collaborator Ellen Hess at Emory doing in vivo microdialysis has shown that Ellen showed that there's abnormal dopamine, extracellular dopamine at rest and after attacks. Shenyang showed that D1 receptors and D2 receptors are upregulated in the mutant mice, but dopamine transporter protein is also upregulated based on striatal extracts on Western blotting. And so, and then Alexander Nelson has done extensive physiology in the striatum where dopamine is one of the major neurotransmitters. But the PNKD protein is expressed, it looks like it's expressed in most, if not all, neurons. So it wouldn't be surprising to me, and Sheniguo showed that it interacts with RIMS1 and RIMS2 to stabilize those proteins. So I would not go so far to say that it's all dopamine. I'm sure that dopamine is involved, but we can't rule out the possibility that abnormal release and glutamatergic inputs into the striatum from the cortex could be involved too, because PNKD protein is expressed in all layers, neurons of all layers in the cortex. So I think that when all is said and done, it may be the case that PNKD mutations might be affecting multiple neurotransmitter systems, but clearly we have data showing that it definitely affects dopamine. Okay.
Now, for the monogenic disease like the PNKD mutation, would the phenotype have a spectrum of variation in terms of the phenotypes?
So the question is, the genetic term would be variable expressivity. In these disorders, so let's just review Mendelian genetics. Penetrance means if you have the mutation, do you have the phenotype? All of the phenotypes I've worked on, talked about here, having very, very high penetrance. Your question relates to variable expressivity. If you have the phenotype, does it always look the same, or can it be worse in some? And I think it depends.
Or would that be affected by other factors such as environmental or?
Oh, absolutely. So there's some of the diseases I've talked about which tend to be more homogeneous. And there are some like Anderson-Tawil syndrome where in the very, very same family with affected people carrying the same mutation, that some are very, very mildly affected and some are very, very severely affected and might die of a cardiac arrhythmia. So there are different amounts of variable expressivity from disease to disease. But if you have PNKD, if you and I both have PNKD, and you're working hard and living the clean life, and I'm drinking coffee all morning and alcohol all night, I can tell you that I'm going to have a much worse phenotype than you. So that's a simple example where, because of the precipitating factor that we know about, patients do drink coffee and alcohol, they're certain to have more attacks than they would if they didn't. And then I think it's true that everything in the middle can happen too. There are epigenetic changes, as you know, that can happen as a result of things we eat, or how healthy we are. Also depends on how we exercise and what we eat, and so I think that all of those things. As a human geneticist, I believe that everything is genetic, but I also believe that everything is environmental.
What about gender difference? Is there any gender
There are
difference?
gender differences in some cases. One that, for familial hypokalemic periodic paralysis due to calcium channel mutations, I don't think we've ever gotten DNA from an affected man with a mutation in that gene that was not penetrant, but 10% of women are non-penetrant for mutations in the calcium channel causing that disease. And I'm sure there are differences with PKD. PKD gets much better as people grow older, but it gets much more better in women than it does in men. So there are clearly differences there, and whether those are hormonal factors or something else about gender, I can't begin to say, but there are always, spectra of differences, and more in some cases, less in others.
Is there any other long-term impact of the PKD mutation? Because if there's an inhibition of the exocytosis, I would assume there are much more consequences coming from that perspective.
Yeah, I mean, they don't, in the case of the periodic paralyses, which is different, some of those patients go on to develop myopathy and a fixed interattack weakness because the muscle can degenerate. In PNKD, we've never, it's so much more rare that we have less, and many of the families are from outside the country and speak languages that I don't speak, so we have less information. We get what we can from our collaborators. But to our knowledge, they don't develop any long-term serious, no degeneration, but nobody's done, taken brains at autopsy and done stereology to look at neuron count, to count neurons in the striatum, for example, or the cortex. Because I'm not aware that there aren't long-term consequences, doesn't mean that there aren't. It only means that nobody has looked and found them yet. But they seem to do quite well, as far as I can tell. And for some of those people, the attacks go away as they get into their middle and later adult lives. And again, I can't begin to. I could speculate. Maybe it's something epigenetic, but I think that's a very interesting part of this whole episodic disease story that nobody, to my knowledge, has made any progress in understanding. So the good news for all you young scientists in the audience is that there will be lots and lots of exciting and wonderful things for you to do in your careers that you can talk about when you get to be my age.
请大家在把握机会，如果需要，我可以帮你听。
Very outstanding talk, thank you. And my question is, since you talk about the dopamine and dopaminergic neuron, I'm wondering whether there is any correlation between paralysis and neurodegeneration, just like Parkinson's disease when the patient is getting old. Thank you.
Well, for the muscle diseases, with the exception of Anderson-Twill syndrome, the mutations are in genes that are only expressed, as far as we can tell, the limits of our ability to measure, in skeletal muscle. Now, of course, if someone is able to exercise more regularly and at a higher level, they may age better. So maybe a muscle disease gene could cause you to have less good CNS function later in life because you can't do as much of the positive stuff to protect our brain health. In the case of Anderson-Twill syndrome, which is highly expressed, it is also expressed it's most highly expressed in the frontal lobes. And this led us to do a study where we brought mutant carriers and their mutation negative siblings in. They have a very interesting and very specific kind of neurocognitive deficit. But we don't have any knowledge yet about natural history and if they develop more severe CNS consequences. So and then for some of the other episodic diseases that affect the brain, like PKD and PNKD, I don't have the sense that any of them get worse as they get older. They tend to get better, if anything else, but we haven't specifically looked at cognitive measures. There's nothing so clear that, you know, everybody in the family who grows to be 70 years old is demented, for example, that we would notice if it was really a big change, but I'm not aware of any changes.
Thank you.
Sounds like people are ready to go for dinner. Who's getting hungry?
I'm the student at Johnson University of Biology Department. And first of all, I would like to thank you for a thorough presentation for us. And my first question is, as you mentioned, the genes that you talk about, what if, is it really happen when people have that gene defect and the people will get the disease, or it depends on the environment?
I'm sorry, I didn't understand. If they have the gene mutation,
yes.
Do they get the disease?
Yeah.
In the diseases I've talked about, they usually do. There are exceptions of non-penetrance, but these are all, you know, as a human geneticist, it's easier, of course, to study big families where 50% of all the children of affected people have the disease. So we've been, you know, biased in studying diseases that are like that. So, and it's been easier to clone those genes than for the more complex stories where there are some genetic diseases like generalized dystonia, which is called autosomal dominant, but where only 30% of gene carriers are affected. And so, you know, so that's a little harder to map and clone in the human genome. Things have changed a lot now with whole exome and whole genome sequencing, next generation sequencing approaches, but we've really focused on the highly penetrant ones because we knew that we could, we'd have more success in finding stuff that we could then begin to study at a more biological level.
And my second question is, since you have found genes that might cause the disease, would it be possible to identify it during the pregnancy?
Oh yes, absolutely. You know, this is a very personal decision for each person, and there's been a lot of discussion and debate about this over the last 3 decades. But it is absolutely true that if you have any one of the diseases that I talked about today, you could, and you don't want to pass it on to a child, you could go to you know, the appropriate kind of doctor in an IVF facility who has the capability of, you can do IVF with the sperm from an affected father or the eggs of an affected mother, and then when you have embryos that are at the 8-cell stage, it's really quite amazing. You can go in and you can pluck one of the 8 cells out, and you can do PCR, and you can say, oh, this one doesn't have the mutation, this one does, and you can say now for implanting embryos in the mother that we're only going to use the ones that don't carry the mutation. And what's incredible to me is that when the child grows up into an adult, they're not missing 1/8 of their body, right? The embryo tolerates this. It's really quite remarkable. And so, it's different for every person. Some of the diseases that I talked about are like PKD. Is extremely easy to manage with low doses of medication in most patients. And I would bet that most parents wouldn't want to go to that kind of trouble because it's not, but for severe neurodegenerative diseases or childhood onset, enzyme deficiencies, there are lots of opportunities for doing pre-implantation diagnosis so that the fertility centers only implant embryos that don't carry the mutation in question.
Thank you.
欢迎 各位师长跟同学们提出问题。I have a question, it's more personal, not related to science. What made you to, as a medical doctor, what made you to choose a career that's more basic science oriented versus practice in the clinic?
Well, I'll try to give the short version. You know, I liked a lot of things, but I was really good at math. And so I got a degree in math in college because I liked math and I was good at it. And I graduated from college when I was very young. And then I had to decide, I thought, well, maybe I should get a PhD in math. But I didn't know if I really wanted to be a PhD mathematician. And in fact, I don't think I had the gift that would have allowed me to, be a really successful academic mathematician. So medical school was kind of a defense mechanism. I thought, well, if I get an MD, I could go into basic science, I could take care of patients, I could go into business, I could work for an insurance company or a hospital.
So you want to have the options.
I saw going to medical school as closing fewer doors. And when I graduated from medical school, I was 22, I was young. Every time I had to make a decision, I didn't really have a whole lot of data. But I was, moving forward, and I was having fun, and I was trying to follow my heart. And so I think it's important to just keep moving forward and do the best, in terms of these decisions. And the worst case scenario is if you end up doing something that you don't like, maybe you decide to take a step back and try something else, right? But, things worked out incredibly well. And looking back at how little I understood at each point when I had to make a big decision, it is amazing to me that, things have worked out. What else are you going to do?
But doing basic research.
Oh, in the basic
really
science. I'm sorry, I didn't answer your question. So when I got to my residency, I was interested in genetics from the beginning because of statistics. Because my emphasis was in statistics, and I thought maybe I would be a population geneticist. So I went to talk to Ray White because he was the chair of human genetics, and he said, "Oh, you're a mathematician. You should work with this guy Mark Lathrop, who's a biostatistician." It was the worst month of my life. It was dreadful, and I decided that I absolutely didn't want to do statistical genetics. So, but it had to do with experiences I had and people I met and talked to, and in my internship, I made my first diagnosis of a new diagnosis of Huntington's disease in a man who came into the clinic, and I started reading about it, and I read Jim Gasella's 1988 paper, I think, mapping. It was the first example of a disease gene that had been mapped to a chromosomal locus using anonymous DNA markers. And as I started to read about that, I was so excited because it was clear that there was a revolution coming. And so I went to talk to Ray White because he was the guy who was leading the revolution. And then I met this 28-year-old patient that was mentioned earlier because I was talking to a medical geneticist friend, and he said, "Oh, I found this interesting family, and I have this patient you might want to talk to." And so it was all, again, trying to, do the best I could, having fun, following my nose, getting, advice from people, and things worked out. Sometimes that surprises me, but it was kind of it was almost a brownie, and it was almost a random walk. I would bump into something and I'd go another way, bump into something, Mark Lathrop, and I'd go the other way. And so I presume you've had some similar experiences.
No, but I would assume, because doing basic research could be really painful and frustrating, and at any point that you might think, okay, maybe I should go back to doing more clinical work rather than doing research because NIH is rejecting my funds and things like that.
No, that's an excellent point. There are many physician scientists who are unsuccessful as scientists. And so when I finished my I was young enough to be totally naive in many ways, and in some ways that may have helped me. I finished my residency and I was a really good neurologist, and it was satisfying to take care of patients and to have them say thank you and have their families, think you were the greatest because you helped their, you know, and helping sometimes meant helping someone to die with dignity. That doesn't always mean that the patient gets better. But then I remember this very clearly. I collected that hyperkalemic periodic paralysis family during my residency. I wrote a grant and I got Ray White to say yes, he would take me in his lab. And so the July 1st, 1990, I started working in earnest to try to map the gene, and 3 months later, in the middle of the night, I was developing a film, and I was using, you know, a Unix machine back then, entering the data into the LOD score linkage program, and you know, I had my first positive result, and it was so thrilling. You know, I loved seeing patients. I loved, you know, talking to the neurology patients and helping them when I can. But when that happened, when I made my first discovery in the middle of the night, it was what I imagined, you know.
That's the moment.
Cocaine might be like, you know. Whoa! I've never tried cocaine, but it was just such a rush that I decided then and there that this is what I wanted to do. So again, you know, for the young people, you got to find what you love, and you have to follow your heart, because if so a lot of clinicians, a lot of unsuccessful physician scientists get into the lab and the negative control is positive and the positive control is negative, and
they tell the PhD to do the work.
And so they say, I'm going to go to a clinic where I can get some warm fuzzies and feel good again. You know, that never occurred to me, and I think it was because at that moment in time I realized that this is so badly what I wanted to do that I didn't consider you know, going to clinic to get a break from. It's true, I've, you know, suffered frustrations in science like everybody else, but I never have regretted that decision, and I never looked back.
Very inspirational. 你们可以问一些就是比较一般的问题，没关系。时间到了哦，那个对，我们这边因为我们这次的活动很感谢，就是我们生科院呢跟我们实生系一起合办，也很感谢大家光临。我们请那个我们陈院长跟我们林主任都说几句话，好不好？The Dean of Life Science.
Okay. Thank you, Louis. Wonderful talk. We think all of us learned a lot from your presentation. Yeah. I also would like to thank our colleague, 研发处的周院长，还有蒋组长把这么好的机会留给生科院刚好在99周年的前夕，能够邀请到2位美国院士跟非研新科的副院士，也是我们的杰出校友。我想这是一个难得机会。那我们这边有一个很好的校庆的飨宴。Yeah. Have a wonderful stay in your trip. Okay. Thank you.
谢谢你。
谢谢。
Thank you, Dr. Louis J Ptáček. Wonderful speech. 非常感谢就是我们那个有这个机会，然后来参加这个2位院士的这个演讲来专题演讲，那应该大家收获都非常丰富那个副院士是我们实生系69人毕业的，60人毕业。那我们现在叫实生，那人叫实科那我们已有荣焉。那非常感谢各位，非常thank you。
谢谢。那我们今天的会，就是我们今天的实生那个会生讲座还有实生讲座就到这边结束。那很感